Hello, everyone, and welcome to the Precision Medicine virtual event. My name is Jonas Korlach. I'm Chief Scientific Officer at PacBio, and I will be presenting on precise DNA sequencing and necessity for precision medicine. Please feel free to submit questions during my presentation, and I will follow up with you via email. Just a quick disclaimer, I would like to note that PacBio products are marketed and sold only for research use purposes, so it's for research use only and not for use in diagnostic procedures. My presentation will have three parts. I'll give a brief introduction and followed by examples of how PacBio technology can be used towards advancing the goals of precision medicine. And then at the end, as a third topic, I will talk about the new Sequel II system. So in terms of a motivation and how far we've come with regard to precision medicine and where the field is going, I'd like to cite two leading scientists in the field and some of their recent perspectives. On the left, you see a slide that was presented by Jay Shinduri, a Howard Hughes medical investigator and professor at the University of Washington, highlighting in the NHGRI roadmap and vision meeting that happened a few months ago that we've come a long way with regard to next generation sequencing revolutionizing medicine, and that's because of the low cost and the very high throughput enabling to sequence literally millions of samples, and his perspective of that this will continue and even expand. On the right on his slide you can see that, and he says it's not unreasonable to think that you could sequence 0.1% at least of all living humans, of the entire human population. So that's, 8.5 million people now. And so this can only be, accomplished by the combination of the low cost and the very high throughput that is afforded by the short read technologies, which have over the past decade really revolutionized medicine and have and really enabled for us to think about precision medicine. And then he pointed out that in addition to those advances that have happened and will continue to expand, there are opportunities. On the left, he has an opportunity for long-read technology such as PacBio sequencing to complement those efforts in generating a much more limited number of high-quality de novo human genome assemblies that are the highest possible quality, telomere to telomere, let's say for dozens of samples and ethnicities, which will serve as a reference to help map the short read technologies and make those more useful. On the right, you see an article from a few years ago by Ewan Ashley, who is at Stanford University, again highlighting how powerful the NGS technology has been in bringing about precision medicine and where we are and where we'll continue to go, and pointing out opportunities, again, for PacBio technology to complement these NGS assays to focus on particular regions in the genome. So they're high complexity regions that are perhaps more difficult to resolve with the short read technologies, and that's where there's a complementary opportunity for long-read sequencing technologies such as PacBio to help provide a more comprehensive picture of a human genome. And as you know, as many of you know, with regard to the technology, it can take advantage of the combination of having long reads, high accuracy, uniform coverage with minimal bias relative to the type of sequence that you're looking at, single molecule resolution, and I'll talk about that in a little bit more detail later, how it is possible to get both long and accurate single sequence reads, and the ability to provide simultaneous epigenetic detection. All of those factors combined aiding in seeing some regions that are perhaps until now more difficult to look at with the short read technologies. And so I mentioned single molecule resolution, and so before I go into the examples, I want to point out that with PacBio technology, there is now a really, we believe, powerful mode of sequencing that combines the long reads and the high accuracy to generate what we call hifi reads. This is done by a process called circular consensus sequencing. So on the upper right, you see the standard template molecule that is being sequenced, a double-stranded DNA molecule in yellow and purple, the two strands. 2 hairpin adapters are ligated to each end of that molecule to be sequenced, which makes the DNA molecule topologically circular. And we call this a smart bell. A primer and a polymerase are bound, and then the polymerase will start sequencing, opening up the structure and keep going around this now circular DNA molecule. And so on the left, you see schematically that it is possible with PacBio technology to sequence the same DNA molecule multiple times over. And you get, on the bottom, you see what we call subreads, which are passes of the polymerase sequencing either the forward strand in yellow or the reverse strand in purple. And then you can wash out the random errors to arrive at a consensus at a highly accurate long read, which we call a hifi read. And the utility of this I will highlight in the following few examples. So I'd like to, in the next part of my talk, show a few examples where these types of capabilities have been used towards informing in the research setting about certain parts of the human genome. The first example is in the area of pharmacogenomics. And so this is due to the fact that people have different responses to medications. It's actually quite a significant problem. It's estimated that about 10% of hospitalized patients experience adverse drug effects. 3.5% of hospital admissions are related to adverse drug reactions, and it's currently estimated to be the fourth most leading cause of death in the United States. And the reason for that is that the underlying reason has to do a lot with genetics. So people have different responses to medications, largely due to their genetic background. This is a figure showing that for the phenotype here in the second row, which is defined as how rapidly the particular patient or the person breaks down a drug, a medicine. And people can be the ultra-rapid metabolizers or average, or they can be poor metabolizers, which means the medicine will stay in the body for much longer. And the distribution shows that there's quite a variety of responses and people. And the top row shows that this can be linked to the genotype, to the number of active genes that you have, the proteins of which degrade those particular medicines. And so it's of great importance to the doctors to know whether a patient is an ultra-rapid metabolizer. And if he would give the standard dose, then the drug would be broken down so rapidly that there would be no health effects. But even more dangerously, perhaps for a poor metabolizer, if the doctor didn't know that that person was a poor metabolizer, there can be potentially adverse effects. And so to highlight the utility of PacBio sequencing to sequence and to understand these genotypes that then ultimately lead to the phenotypic variation, Stuart Scott, who is at the Icahn School of Medicine at Mount Sinai and also at Semaphore, gave a presentation a little while ago entitled Amplicon Smart Sequencing Applications in Human Genetics, introducing one of those important genes that's called CYP2D6. It metabolizes about a quarter of all current Western medications. But he also shows in this slide that the genes are very complex, and it's challenging to do this with current techniques because they are neighboring pseudogenes. The gene is highly polymorphic, meaning there are many different alleles and many different forms and variations of the gene. And in his talk, he nicely highlighted the progression that has been made from when he started this work in 2015 on the first instrument, the RS2 that he had. Trying to span, let's say, a 5 or 6 kilobase gene was still a bit of a challenge, whereas in 2018, with the sequel system and the version 3.0 chemistry, sequencing chemistry, you see on the right with the blue that there's very even coverage now that allows you to fully sequence the 6,000 base pair amplicon that represents the full-length CYP2D6 genes in high multiplex. Here he multiplexed 48 samples in one SMRT cell, and gets an average depth of 1,300 folds, so way more than you would ever need. And he then highlighted that there is a very high accuracy and precision, so the triplicate variant haplotype concordance was 100%. This picture here on the bottom shows an IGV view of the entire CYP2D6 gene. That's about 6,000 bases in this view, and you see 2 blocks of rows. The pink lines are the forward strand reads, the blue lines are the reverse strand reads, and the two blocks delineate the two haplotypes, the two alleles. So you have one copy from the mother and one copy from the father chromosome. And the vertical lines represent the variants relative to the reference genome. And so you can see a couple of things. You can see how clean the data are. So these reads are essentially perfect because there are no colors in between them. And that's a power of the hifi read, the long and accurate read. And the second thing you can see is that it is how easy it is to phase the variant information. So you can see in this example, all the vertical lines, the variants relative to the reference that represent heterozygous SNPs, are phased together on one allele. So all the variants are coming from one of the chromosomes, let's say the mother chromosome, whereas the father chromosome is fully concordant with the reference. And so doing this work and comparing it to previously genotyped samples, here's 96 samples, he found that the PacBio data were fully concordant, but not only that, in addition, he found that for about 10% of the samples, he had to either revise or discovered completely novel variants and CYP2D6 diplotypes for the sample that he didn't know about before. And to stretch the technology a little bit, he looked at another pharmacogenomics important gene, that's 11,000 bases, and very similarly, and here comparing the PacBio with SQL and the version 3 chemistry, you can see again that all the variants are very nicely called and phased. And for example, here showing in addition a 25 base insertion that, so not just SNPs are being detected, but also insertions and deletions. And you see all the reads basically show that this is a 25 base pair insertion that only is present on one of the haplotypes. The second example that I'd like to highlight today is in the area of bipolar disorder and schizophrenia. And this is from a talk recently given by Janet Song, who works in David Kingsley's lab at Stanford University. She gave an updated talk just recently last month at the Smart Leiden conference in the Netherlands, looking at the utility of PacBio technology to help identify the causes of this important disease. You see on the bottom, it is a big problem, affects over 3% of individuals worldwide, and is in the top 25 causes of disability. But despite many years of study and GWAS assays and so forth, the causative variant of this for this disease is still unknown. They then looked at, I can't go into the detail, but the talk was actually recorded and you can find it on our website. So if you want to know more detail, they found a region for calcium in the intron of the calcium channel that was highly variable. And we're using PacBio sequencing of initially 16 individuals, identified that there was this region that was very variable and had a repeat variant. So by Southern blots, this was initially identified and then resolved at the sequence level with PacBio sequencing. And there were about 50 detected variants on the 30-member TEEF. So this is shown here in the publication that came out since then. So if you would like to know more about that, I refer you to this paper. And in this colorful figure E that you see there, each row is a different patient. And you can already appreciate just by the variety of the coloring that there's a tremendous amount of variation in this particular tandem repeat region that was characterized using PacBio sequencing and can be unequivocally resolved in terms of the structure and the sequence composition of this human calcium channel gene that they're now looking at further. And she gave an update at the Leiden meeting about the potential significance as both enhancing or repressing the effects that may lead to bipolar disorder and schizophrenia. There was also a press release on the right entitled Hidden DNA Sequences Tied to Schizophrenia and Bipolar Risk, highlighting how the PacBio technology can be complemented with the high throughput and cost-effective short-read technologies to get a more comprehensive view of the genome. The third study is in the area of HLA and HLA typing. There was a recent paper that I was very excited to see by Anthony Nolan and by the Anthony Nolan Institute, Steve Marsh and Nima Mayer on the right, leading this work, providing a high-resolution HLA typing and associating it in a retrospective study with the patient survival rates. So they show these very dramatic Kaplan-Meier plots showing that when you actually match the patients with regard to the HLA match, again, this was a retrospective study, there is a very significant increase in the overall 5-year survival rate compared to thinking that you had matched them, but then with full-length HLA gene sequencing showing that this was actually not the case. And then finally, in the area of going from targeted sequencing to having a look at the entire human genome, we collaborated with, as you can see on the right here, a large number of bioinformatics experts in the community and posted a preprint a few months ago looking at the utility of the CCS reads to inquire on the whole genome scale about some of those regions. If you want details, I refer you to the preprint, which is publicly available, but very briefly, we looked at one of the best studied and characterized genome in the bottle benchmark samples, and the reason for that was to evaluate how well we did relative to what is best known as the biological truth. And the CCS reads were able to then improve on the benchmark data call set of the Genome in the Bottle initiative. Again, CYP2D6 is shown on the left here, phasing and resolving the haplotypes unequivocally, and overall an estimated 2,400 benchmark mistakes were corrected. The benchmark was expanded by about 150,000 variants, and significantly, in view of the topic today for precision medicine, adding about 5% more variants in what's considered the medical exome. And so just to highlight that a little bit more, one of the figures in the paper shows that there are a number of genes, and they're listed here, and characterized previously by Birgit Funke and colleagues in this paper on the upper right, that because of the presence of pseudogenes and highly homologous regions, all those genes are very difficult to resolve completely with the short read technologies. But our analysis indicates from this paper that the large majority of those genes are now accessible through the long and accurate PacBio read sequencing. Even some of the most difficult regions, like the SMN1, SMN2 genes, are near reach. These are very extremely homologous regions, pseudogenes that have a very high percent identity. And now with the hifi reads, they're coming in reach to being even those types of regions being resolved. So then in the last few minutes, I then would like to just give an update on the latest capabilities that aid these types of examples and more for helping fulfill the goals of precision medicine. And the Sequel II system, in the context of how at PacBio the technology has evolved, this is a slide showing the throughput evolution over the commercial lifetime of the company, so about 6 or 7 years. We have built on the foundation of the high quality that I mentioned in the beginning of having long reads, having high accuracy, minimal bias. We have been able to then build up that foundation with regard to throughput, increasing the overall throughput by well over 10,000-fold by now with the introduction of the Sequel II system, increasing the average read length by over 100-fold and so forth. So then in April of this year, we launched the Sequel II system, which has a smart cell, a chip that has an 8-fold higher feature size. So that translates directly to an about 8-fold increase in the overall yield of sequencing. And this is what we call the Smart Cell 8M. And what that translates to is the same kind of performance, equivalent performance with regard to read length, accuracy, lack of bias, and so forth, but now with the higher throughput, translating to reduced project times and lower cost and so forth. And so how the HiFi reads now look in the context of the Sequel II system, I showed this slide before. This is a representative example of the yield that you get. So you can see that for these types of amplicons in medium range, so maybe up to 20 kb DNA inserts, the read length distribution has completely shifted to the right, where most of the reads are now on very long reads. Half of the reads in this particular example are well beyond 100,000 bases. In fact, for this particular example, the overall throughput was 320 gigabases. Half of the data was in reads that were greater than 167 kilobases, about 4 million reads. So overall, the raw throughput of the system is up to 400 gigabases, the average read length up to 100 kilobases. And so then how many passes do you need on the Sequel II system to get to a HiFi read? So this is shown in this graph, and we actually made some improvements in this regard relative to the previous Sequel system. So now it takes only about 3 passes to get to Q20, or 99% accurate reads, and it takes about 8 passes to get to Q30, or 99.9% accurate reads. So that in the end, then, you end up with, depending on the insert size and the particular experimental design, you get between 1 and maybe 3 million of HiFi reads, highly accurate Q20 or greater CCS reads. The read distribution of CCS reads is shown on the right. So in this particular example, the average insert size was 12,000 bases. So it translates to about 12 gigabases of HiFi reads, about 1.9 million bases altogether. And the average single read accuracy now is 99.8%. But you can see that the mode of the distribution is actually much closer to Q40. So that's actually 99.99%. And the same minimal GC percent and sequence context bias holds as you've known for the PacBio technology over the years. So this, we've just launched a system and it's coming online, and the first service providers are now providing service at Hudson Alpha, at Mount Sinai, and at the University of Maryland. There is a recent preprint that I want to point your attention to if you want to learn more about high fidelity long reads from the Sequel II system, in collaboration with Evan Eichler, also Howard Hughes medical investigator at the University of Washington, comparing both with regard to variant calling as well as de novo assembly, the new HiFi approach, comparing it to the more traditional continuous long read or CLR approach, looking at specific regions of the genome, such as segmental duplications or pericentromeric regions. And I'd like to encourage you to check out this new preprint if you're interested in more detail. So with that, I'll close, and thank you for your attention. And I want to close by pointing you to a recent webinar that happened last week, where 3 early access Sequel II users described their experiences with the new Sequel II system. The recording is now available on our website at picb.com/videos. And so if you're interested, I invite you to take a look at that and how the researchers have applied the Sequel II system for their particular research interests, including the topic that was the topic today, precision medicine, but also going far beyond that. So with that, I'd like to close. Thank you for your attention. And as I said before, if you have any questions, please reach out through the questions, and then I will answer you by email. Thank you, and have a good day.